Hello, good afternoon, everyone. I hope you can hear me now quite well. I first want to welcome you to our biostimulant webinar. This webinar is organized in the framework of the Straits Committee project called Cross-Border Knowledge Exchange to advise growers in their choice of using biostimulants to cope with future challenges. 3 partners are involved in this small project. We have Viaverda in Belgium, Unia in France, and NIAP in the UK. I also want to say quickly that it can be that we go a bit over time at the end, so it's like 10 or 15 minutes. So it can be for 10 past 4 or 15 past 4 when we will end this webinar. Okay. First, some general info to follow this webinar. As you can hear, this webinar is organized in English, but we have some subtitles for the Dutch and French-speaking people. So I will first give a word in Dutch. Hallo iedereen, goedemiddag. Als jullie graag live ondertiteling hebben bij deze webinar om makkelijker het Engels te begrijpen, dan kan u eigenlijk bovenaan het scherm op het icoontje caption of ondertiteling klikken. En daar gaat u de optie vinden om de taal eigenlijk te kiezen. Dus daar kan u kiezen tussen Nederlands en Frans. Sorry. Ali, can you say it also in French? You're still muted.
Good afternoon, everybody. Yeah, and bonjour tout le monde. Merci pour votre participation sur ce webinaire sur les biostimulants. En tout cas, la langue qui est prise en compte dans le webinaire, c'est l'anglais pour les présentations. Par contre, on a la possibilité de faire une traduction automatique sur le système Teams. Donc, pour ce faire, il faut aller sur Caption, comme vous pouvez le voir ici sur l'écran, et choisir justement la langue qui vous convient à tous pour que vous puissiez comprendre en temps réel les présentations. Donc, ça, c'est pour les captions, la langue. Et puis, il faut savoir aussi que vous avez à côté, à un autre endroit ici, Q&A, c'est pour poser des questions sur le chat. Justement, si vous avez des questions qui vous viennent à l'esprit au fur et à mesure des présentations, n'hésitez pas à les poster sur, ici, l'onglet Q&A. Voilà pour moi.
Okay, thank you, Ali. So Ali already said it in French, but I will repeat it in English. So normally the Dutch-speaking people have their Dutch captions. So you have the option during each presentation to ask some questions in the Q&A box. So there you can find the logo on the upper part of your screen. It's called Q&A. And if you have a question, please ask it over there, and then we will look at the questions after each presentation. Then a small overview of the program of today. So the webinar is divided in 2 parts. The first part is more scientific oriented, and the second part more practical oriented. We will start with our first guest speaker, Patrick Dujardin from the University of Liège and Gembloux ABT, and he will talk about placing plant biostimulants on the EU market status and issues. Then, as our second guest speaker of today is David Barton from Prospero Partners, which is a member of ABIC, and he will say a bit more about who ABIC is, what they do, and what their key issues are. Then Ali from Unia will say something more about the mode of action of biostimulants. In our second part, Els Pauwels from Viaverda will say something more about the different types of biostimulants and give some trial results. And then at the end, Matt from NIAP will close this webinar by presenting our Bio4Safe Biostimulant webinar, a free online database. Before we start with the actual webinar, we want to ask you a few questions. So I will go to Mentimeter for that. I'm just going to share my screen again, like this. So for this, I want to ask you to take your smartphone with you. To to participate to this poll, you can scan the QR code on top of the screen. And then, so I give you 1 minute to do this, to open the poll on your smartphone. You can also, if you don't have a smartphone, there's also an option to go to the website of menti.com, and then you can use the code on top of the screen. That's also an option. And as you can see, we have a first question. This question is, which country are you from? We have Belgium, France, and UK, the three countries that are participating in this project. And then we also have the option other. So we can see that a lot of people are filling in the poll. That's nice to see. Thank you for that. Okay. Okay, I can see that still people are filling in or answering to this question. Yes, and they can still do it, but to not lose too much time, I will go to the next question of our poll. And this question is, what's your role? Are you a grower, a researcher, a student or teacher, a policymaker? Are you from the biostimulant industry, an agronomy advisor, or other? See that the answers are coming in. At this moment, most of you are agronomy advisor or researcher, but we also have some growers, student or teachers, policymakers, and some people from the biostimulant industry. So this is very interesting for us to see what you are doing. Okay, then I will go to the next question. If you are a grower, which crops do you manage? Multiple answers are possible. We also have the first option, field crop plants, cereals, potato, vegetable, fruit and berries, ornamentals, garden, park and urban green management, other, or the option, I'm not a grower, because we saw that not everyone is a grower. We only have a small percentage growers, but it's interesting for us to know if there are growers present, which crops they grow. So please also, if you're not a grower, fill it in that you're not a grower. Okay, I think we can go to the next question. Then again, a question for the growers. Which challenges are you experiencing? Nutrient stress, drought stress, waterlogging, heat stress, cold stress, salt stress, or other stresses? Or also again, the option, I'm not a grower. We want to know this because biostimulants are known to be used in conditions of stress. So for this, we want to know which kind of stress you're experiencing on your company. Okay. At this moment, if you are experiencing stress, it's mostly nutrient stress, followed by drought stress and waterlogging. Also very interesting to see. If you have other types of stress, you can also type it in the Q&A if you want. Okay, next question. Do you use biostimulants to address these challenges if you are a grower, of course? Yes, I'm using biostimulants. No, but I'm planning to use them in future. Or no, I'm not planning to use them. Answers are still coming in, so that's good to see. Thank you. I will just wait a few seconds. Okay, and then I will go, I think, to the last question of today. If you use biostimulants, which products do you use? So normally for this question, you have the option to type your product that you are using at your company. Okay. If you are not using products, you can indeed answer none. But also the products are coming in. We see Trichoderma, Asperello, Maxicrop, Silica Power, Composti, amino acids combined with seaweeds. But most people are not using them, as we already saw in the previous questions, of course. Okay. Answers are still coming in. Very interesting to see. Okay, I will close this question, but I think you still will have the option to fill in your product that you are using. So this was the last question of my poll. For now, I will give the word to Matt first. Thank you for filling.
Yeah, thank you everybody who answered these questions. We really like to know who is attending the webinar, and then some funders of ours like to see that as well. And it's really good that you have already been expressing your views on the different things that we're going to discuss today. And we're going to kick off with presentation with Professor Patrick Dujardin from University of Liège, and he's going to look at or tell us a few things about the new regulatory aspects of biostimulants. So, Patrick, the floor is yours.
Thank you very much for this introduction. I'm professor of plant biology, of plant physiology, of plant nutrition, but indeed I've been asked to talk about regulation, which, by the way, raises scientific questions about definitions, of intended uses, and I'm happy to discuss with you a couple of questions. We have a new regulation in Europe since 2019 on the placing on the market of fertilizing products, and this regulation has come into force in 2022. We had a regulation in Europe for the placing on the market of fertilizers, which are defined as suppliers of nutrients. But in this new regulation, we have not only fertilizers, but we have fertilizing products, which are more than fertilizers, because we have also other products with other functions than just the supply of nutrients, and biostimulants, of course, is one of those new categories, functional categories. So what's really new in this regulation, I would say, is that we have products aiming not only at supplying nutrients, but also at improving nutrition efficiency. Another important aspect of this regulation is that it aims at using an increasing number and diversity of raw materials coming from industrial byproducts or wastes. Basically, this regulation should be understood as a contribution of what we call circular economy. So we have a new regulation for introducing new product functions, but also new component materials on an EU harmonized market, of course, because we had already national fertilizers regulations. We already had biostimulants on the markets of European countries. But of course, the aim here is to have a harmonized system at the EU level. The main point to understand, I believe, in this regulation is that we have, in fact, two sets of categories. We have one product function categories series, and we have one set of component material categories. So in order to place on the market a fertilizing product, you have to define which is the intended function of the product. That means that you have to say which PFC, product function category, you're talking about. And of course, today we will address more specifically this product function category 6, plant biostimulants, which is really something really new in the regulation. But also something new that we will talk about is regarding the component materials categories, is the component material category 7, microorganisms, and we will see which kinds of microorganisms we are talking about. Okay, so when you place a fertilizing product on the market, it will be defined by a function, but also defined by the component materials that you will use for making these fertilizing products, delivering this intended function. And for each of these categories, you have specific requirements. We will see that you have European harmonized standards allowing you to define the characteristics of your product. These are different topics that I will address with you in this talk. So product function category 6 is plant biostimulants. And we have a definition, of course, of this category from the regulation. And it's important to have a good understanding of this definition. You see that it is a product, the function of which is to stimulate plant nutrition processes independently of the product's nutrient content, with the sole aim of improving one or more of the following characteristics of the plant or the plant rhizosphere: nutrient use efficiency, tolerance to abiotic stress, quality traits, or availability of confined nutrients in the soil or rhizosphere. What is really worth underlining in this definition? As you see, the biostimulants are defined not by what they are, but by what they do. And more specifically, they are defined by claims, which are the intended effects of those products on crops, which are these agricultural functions that the farmer is interested in, of course. That's very important to understand for the later understanding of how this regulation will be implemented. So Europe was the first to have such a broad, widely accepted definition for Europe, leading the way to other regulatory advances. And we can also mention this definition adopted by ASO at the OECD level, And if you read the definition, you also find that biostimulants are defined by those claims: improving nutrient availability, uptake or use efficiency. The exact wording might be a bit different, but of course it's the same. Tolerance to abiotic stress and consequent growth, development, quality or yield. So biostimulants are defined by claims. But then we have the question of how we can validate the claims. For placing a fertilizing product on the market in general, a biostimulant in particular, you will have to validate the claims, which means as a company that you need to have the data supporting the claim. No data, no market, I would say. But then we have at the EU level technical specifications and EU harmonized standards, which will define what we mean by safety, by agronomic efficacy, how to demonstrate efficacy, what kind of protocols should be used. These are all implementation standards that will be essential also for the placing on the market of these biostimulants. The system is a system of conformity compliance check. And that means that you have in Europe certified notifying bodies that will check the data supplied by the applicant and checking that the data indeed will comply with the standards, with the requirements set by these standards. Next, you will have a self-certification by the manufacturer, which will issue a certificate of conformity, providing assurance that the product sold to the end user indeed does satisfy the requirements of the product. The next step then will be the CE marking. You are quite familiar with the CE mark on all kinds of products on the EU market, which means that this product does satisfy the conformity assessment and the standards underlying this assessment. We also have since recently a digital labeling, which is now enforced for fertilizing products. We have provisions for physical and digital labeling for the operators and end users. But the question I would like to address with you is it possible to standardize biology, agronomy? We know that the effects that we can observe in the field, the response of the crops, of the plants, of course, can be much dependent on the soil conditions, the environment. And this is exemplified by this figure. It comes from a commercial leaflet. And what you see here is that you have 73 field trials in winter wheat, using seeds coated with a biostimulant based on humic and fulvic acids. And yield is measured, and you can see that you have yield increases for most of the trials, but you can also have yield penalties for a minority of the trials. But that's the reality, and we have to deal with. The response, of course, it might be expected, but not necessarily to such a level. You expect, of course, variation in the response of the plant to the biostimulant in the particular receiving environment. Still, the regulation says that the plant biostimulant shall have the effects that are claimed on the label for the plants specified thereon. So that's basically the challenge to all of us, scientists, growers, companies, regulators. We have to make the system as efficient as possible. We have standards at the EU level. I would like to raise a couple of points, which for me are a bit problematic. Probably we have to work on it. One difficulty I see is the grouping of crops that is indeed proposed by the standards. For validating the claims, we have only three groups of crops: arable crops, woody crops, and vegetables and herbs. But of course, if you demonstrate that something is true for apple trees, it's a bit difficult to extrapolate to other woody species, pears, or even walnuts. So that's a difficulty, I believe. We have sometimes a poor definition of soil types. Physical and chemical properties of soil types, which will also influence the response, of course, to the biostimulants. For the statistical analysis, high p-values are acceptable. That might also create some problems, I believe. And a problem I see as a scientist that, and in contrast with other systems, like for plant protection products, yes, it will be asked to the company to provide data, field trial data, but there is no obligation to pre-register the trials. So my question is a bit provocative, I have to say, how to avoid the cherry picking of the best trials by companies for the compliance check. We have to ensure the safety of fertilizing products. We have to understand that it's not a case-by-case assessment like for plant protection products. It's based on compliance to safety standards, which are set by the fertilizing products regulation. For instance, the maximum levels of contaminants, cadmium contaminants in phosphate fertilizers, for instance, or Escherichia coli and Salmonella in bacterial biostimulants. We have also rich regulation, so for the placing on the market of chemicals, which also does apply at least to some categories of component materials that might be used for making biostimulants. And we have the special case of microbial biostimulants, which I will discuss into some more details now. So microbial plant biostimulants are a product function subcategory, which is identified by a positive list. That means that we have under this category defined as a microorganism or a consortium of microorganisms. We have a reference to a positive list in the Annex 2 of the regulation. And this positive list today is extremely limited. It only contains Azotobacter, mycorrhizal fungi, Rhizobia, Rhizobium, and Azospirillum. That is the initial positive list set by the regulation. But there is today quite extensive work to expand the list, but of course also to agree on the criteria for expanding the list while ensuring the safety of the products. If you want to expand the list of the microbes under this regulation, you have general criteria, general for all so-called delegated acts which allow to modify the regulation. You have to show, to demonstrate that there is a potential to be subject to of significant trade on the internal market. Okay, but you have to justify why a national system is not enough, why you should upgrade this to the European level. But of course, you will also have more specific criteria to verify which strains of the additional microorganism are safe for health and for the environment as well as agronomically efficient. And we have a couple of criteria set by the regulation, but how to verify this, how to measure, how to implement these criteria is something which is now being worked at the level of the Commission, and there are different contracts now ongoing for moving forward and expanding this positive list of microorganisms. But what if a substance or a microorganism protects against diseases, so is a plant protection product and biostimulates plants? This question is relevant because we have many examples of either substances or of microorganisms which have both activities. They protect the plant against diseases, but they also have biostimulation effect defined by the claims that we have seen. And that's important because we have separate regulations and the way they work is completely different. And I want to underline some of these differences. Compliance check for biostimulants products, basically with self-certification by the manufacturer, while for plant protection products, it's really a case-by-case authorization of active substances first and then of the formulated products. We have the CE marking system working with biostimulants products, which is not working for plant protection products. Safety requirements have been defined for biostimulant products with maximal levels of contaminants. I explained to you this. we have really a case-by-case safety assessment for plant protection products. And one important difference also is that for biostimulant products, there is no mandatory characterization of the modes of action of the biostimulant. While this is really essential for plant protection products, where you really need to define the primary targets of the active substances, they have to be characterized. Of course, for the company, it's extremely important and useful to understand enough about the modes of action of the product, but the regulation does not demand a clear demonstration of the mechanisms of action of your biostimulant. So how to separate biostimulation and plant protection? Because we have two regulations, sometimes one product has both activities. But what's important to understand is that the regulation talks about a function, which means that products with one or more functions, one of which is covered by the scope of the PPP regulation, Plant Protection Product Regulation, or plant protection products falling within the scope of that regulation. And these products should remain under the control developed for such products and provided for by that regulation. So, but how to understand this? Does that mean that any biostimulant with pesticidal or biocontrol activity should be de facto excluded from the FPR regulation? No. The answer is clearly no, because we talk about functions and not about activities or effects. And the function describes the intended effect of a formulated product in specified conditions of use. It is not any biological activity of the product constituents. So you might have a microorganism with different activities, both for biostimulation and for control of diseases, but it will depend on how you design the product for intended function. And then it will be possible to place on the market this product as a biostimulant. To conclude, I would like to challenge the concept of plant health, bridging plant nutrition, biostimulation, and protection. I would start from the definition of plant health set by the World Health Organization for humans, for human beings. Health is a state of complete physical, mental, and social well-being, and not merely the absence of disease, of infirmity. You see that health is more than the absence of disease. If you read a draft regulation on soil health, you see this definition of soil health, the physical, chemical, and biological condition of the soil, determining its capacity to function as a vital living system and to provide ecosystem services. Again, soil health is a capacity to fulfill functions. Okay, it's not just the absence of a disease or damage or whatever. And what about plant health? Quite surprisingly, if you see how plant health is defined in the EU regulation, basically it's a negative definition. It's the absence of diseases, which means that to combat these diseases, you use plant protection products. If you want to improve health, you need plant protection products or quarantine measures, etc. Of course, for the limitation of the entry of new diseases on the European territory. So what I would suggest is to move to a positive health concept, which means that health would be considered as a capacity to carry out its physiological functions to the best of its genetic potential. You see that it's more than the absence of diseases again. In that case, plant protection products, of course, will be useful, but biostimulant products also will be instrumental, I believe, for increasing this health concept, the health of the plant according to this positive health concept. So plant care is definitely more than plant cure. And this is my conclusion. Time to conclude. There is this Ayurveda sentence saying that when the diet is wrong, medicine is of no use. And when the diet is right, medicine is of no need. I would suggest that we could maybe translate this to crop nutrition and crop protection, saying that when plant nutrition is wrong, plant protection is of no use. And when plant nutrition is right, plant protection is of no need. I don't think we can go that far, okay? But still, I believe that connecting plant protection and plant nutrition is certainly very helpful for sustainable agriculture and also for revisiting globally plant health. I thank you for your attention. Of course, I will be happy to answer any questions. I'm not sure that we'll have much time to do it right now, but up to the chairman or chairwoman.
Yes, thank you so much, Patrick. That was very interesting. We got one interesting comment that I'll just put out there, and that is basically summarizing that if we don't know or we don't need at the moment mode of action for biostimulant registration, that makes a lot of people very skeptical. And I think I would agree. I think the mode of action should be there, at least some mechanisms, maybe not as, I don't know, what's your point?
Yeah, I fully agree. I fully agree that a difference probably will be whether the data on the modes of action will be shared or not, because that will make the difference probably. The fertilizing product regulation has no provisions for data protection. Okay, that's an issue. That means that if you make the mechanism of action mandatory in a dossier, we have to guarantee also that there is some possibility for the companies to protect the intellectual property, because there are few ways for protecting the intellectual properties of these biostimulants. That's a difficulty. So many patents are bear on the manufacturing process, etc. But still, it's a challenge. There is a difficulty there to be solved. But obviously, and of course, as a professor of plant physiology, I believe a lot in the usefulness of understanding as much as possible the modes of action, because that's also the way we will be able to deliver a sound advice to the farmer about the conditions of use of the product, of course.
Yeah, we have two very quick questions. Thank you for this, Patrick. One is, are the field application of biostimulants limited to food production? No, they're not. You can use biostimulants in any production of, even in your own back garden. And then this one, I'm not quite sure. What is the difference between biostimulants without authorization number and biostimulants with an authorization number? Now, are biostimulants actually have an authorization number across the EU at the moment?
Not as far as I know. The authorization number, there is no, to the best of my knowledge, there is no register of products, but this is probably a question that David later can maybe answer from EBIC, but to the best of my knowledge, there is no authorization number. I will try to answer the first question. No, definitely biostimulants are not limited to food production. There are applications in tree nurseries for promotion of rooting, and even for the public greenery and gardens, you already have some biostimulants, seaweed extracts included in some fertilizers applied for orchids cultivation, etc. So you do have, not necessarily at the European level, and that's important to stress that we have a few products coming now under the European regulation, but we had quite a significant number of products on the national markets and in all sectors of plant production.
Okay. Thank you. That gives me a really good segue to David Barton from Prospero and represented European biostimulant industry today. He's going to talk about what are the key issues in biostimulant industry, maybe touch on registration again. So, David, take it away.
Thank you, Matt, for that introduction. So, good afternoon, everyone. Delighted to be here today. Representing the European Biostimulants Industry Council, or EBIC. So I'm just going to explain today a little bit about who we are, what we do, and how we're shaping the future of the biostimulant industry. So just a little bit of history, I guess. EBIC is essentially the voice of the biostimulant industry in Europe. We were founded in 2011, and to basically provide a unified platform for stakeholders across the biostimulant value chain. So we started off with 10 members. Today we're 70 members, ranging from micro businesses right up to multinational corporations. So you can see from our membership or an infographic, I suppose the weighting between different membership classes is quite equal. So it is a good representation of the industry, and as a consequence, you know, we get a very comprehensive understanding of the market and the needs of all stakeholders. So if you want to find a full list for membership, feel free to go onto our EBIC website, which is biostimulants.eu. So our vision is clear: sustainable agriculture supported by the global adoption of plant biostimulants. We're committed to promoting biostimulants as essential to advanced food systems through education, advocacy, and support. Our values, such as sharing expertise, enabling innovation, encouraging collaboration, and demonstrating leadership, are the core values that guide everything that we do. In terms of timelines, just to give you an idea of what's been happening since our genesis back in 2011. As I said, we started with 10 companies. By 2012, we sponsored the first World Congress on Biostimulants, so effectively putting the sector on the global map. In 2013, we launched our code of conduct for our membership, which sets ethical standards for the industry, and the objective was to reinforce trust and transparency. And I don't mind sharing that we lost some members in the beginning when we made this a condition of membership, but we do believe in bringing products to market with integrity, so it's an important cornerstone of our membership policy. Fast forwarding then to 2016, biostimulants were officially included, as Patrick alluded to, in the fertilizing products regulation. And by June 2019, this was adopted and formalized within the EU legislation. So it wasn't until July 16, 2022, practically 10 years to the day from EBIC formation, that the first EU fertilizing products could officially enter the single market under the FBR. So this was a landmark achievement after years of advocacy. So in terms of EBIC's part in this milestone, we played a critical role in shaping the regulation. And as a consequence, we now have clarity and credibility because biostimulants are officially recognized and we have a legal definition, and they're now included in a broader framework of plant nutrition and soil fertility. So this really has reinforced biostimulants as a credible, safe, and effective crop input class. So first 10 years of our life as an association, it was really focused on removing regulatory obstacles. So we have the FBR. It's not perfect because we had a certain legislative window and a certain space of time before the Commission changed to get it across the line. So there are still elements that we still advocate on a regulatory basis, but we also look into the future, and to stay ahead, we have a very clear strategy for that. We're focused on enabling market harmonization in Europe. We're looking to drive global business development and strengthen the position of EBIC as a thought leader and cultivate both innovation and promote sustainability. So we're very much a bottom-up association. So these strategic priorities really do reflect our membership and needs and wants, and will guide our efforts over the coming years. So as I said, EBIC isn't just about advocating for regulation. We're also focused on building the market for biostimulants by tapping into opportunities like cap incentives, aligning with ESG priorities, and also exploring insurance rebates as well. So these effectively can create pull factors to drive market demand. And what we're trying to do here really is make biostimulants a mainstream input class and indispensable to sustainable agriculture, because we want them to be seen not as a nice to have, but as a critical foundational element of that farming system. So we really have kind of shifted towards building that demand, not just in Europe, but globally. And we're actively looking to create those pull factors to drive faster adoption of biostimulants. That being said, to fully unlock the potential of biostimulants, we need enabling policy frameworks. So we have, I suppose, we took the opportunity with the change of commission to publish a policy manifesto. So this is really our blueprint for supporting biostimulants in the future. So what we're looking to do is call for policies that empower farmers, reduce regulatory hurdles, and foster climate resilience. So we're pushing for incentives that just make it easier for farmers to access biostimulants and adopt them. And if you want to find more about our manifesto, I will be making this presentation available afterwards, so there is embedded links, but you can also use this opportunity to scan the QR code on the bottom right to access our manifesto now. In terms of fostering collaboration and thought leadership, about 3 years ago, we established our own event for EBIC members called the EBIC Summit. So this really is a platform for key players across the value chain to come together, collaborate, share ideas, understand challenges, and speak about opportunities to overcome certain challenges. So it really is an event that we're looking to animate the value chain from farm to fork and widen our discourse beyond just the biostimulant industry, because we believe having a diverse network creates opportunities and will, I suppose, help position biostimulants as a valuable input class that can address other needs across the value chain, and that's so. It is a member-only event. Our next one is in June 12th in Brussels. So I would very much encourage you to join EBIC before then, if you wish to attend, and happy to see you there. So as we grow, our focus is expanding to engage with C-suite leaders in industry, not just from the biostimulant industry, as I said, but across the value chain. So traditionally, we've been seen as regulatory allies. So we're now really looking to position EBIC as a strategic partner. And our goal is to really, as I said, create those pull factors and build on the strong regulatory foundations that we've established. So to do that, we're looking to break down silos, foster connections, and help members tap into new opportunities and that broader network. As I mentioned with the EBIC Summit, that's one platform that we're looking to do that, but we also co-host the Biostimulant World Congress, and that is another useful platform to get these connections and networks developing. So speaking of pull factors, the Common Agricultural Policy Eco-Scheme incentives are a great example how incentives can drive the adoption of biostimulants. So this is a financial incentive, and we saw the impact of this firsthand in Greece, where I think they were offering incentives of 30 euros per hectare for arable crops and up to 60 euros per hectare for high-value fruit and veg crops. So it's no surprise that this scheme was extremely popular and became oversubscribed. But I think the demand for such schemes is evident, And I think given the right support, you can see that farmers are eager to adopt biostimulants and invest in more sustainable practices. So this isn't just limited to Greece, and it's something that we're advocating quite heavily from an EBIC point of view to try and influence national strategic plans in this regard. Equally, I think at the EBIC summit, two important themes surfaced. One was to do with food companies and their ESG and environmental goals, and also with crop insurers. They had a big need in terms of increasing climate resilience or crop resilience to climate change. And so we had some really good discussions on that. Food companies are looking to reduce their scope 3 emissions, particularly from fertilizer use. And I know of one company that I talked to where 50% of their scope 3 emissions were due to fertilizer use. So biostimulants can play a role here by improving nutrient use efficiency, lowering fertilizer inputs, and consequently cutting carbon footprints. Crop insurance people, it was, I think they were quite intrigued with biostimulants, and they saw them as offering a way to increase crop resilience. And if the data is of a sufficient quality, then they were happy to offer insurance rebates for farmers who adopt them, and they had no problem making such recommendations. So these are some of the outcomes from our summit in June, and I think it's just goes to show those two examples of the opportunities in creating a diverse network and sparking opportunities for the use of biostimulants, and how can we meet those opportunities. So the insurance company one is something we will pursue, because as you know, and a lot of you might be familiar with this graph, that abiotic stress remains one of the biggest challenges in agriculture, and accounts for up to 70% of yield losses, which you can see there in orange, depicted in orange on the bar chart, whereas biotic losses account for less than 10%. So that's quite a disparity. So if biostimulants can reduce crop insurance payouts even slightly, it could have a big impact. So as a consequence, in terms of our advocacy, where we understand data-driven advocacy is more compelling. So we are looking to take a role here and build data sets from our membership. So it's something we're doing, but it's not going to happen overnight. It's very much a process. It's going to take some time, but I suppose just to give you a timeline, we started this back in 2023, where we surveyed our members with respect to nutrient use efficiency claims, and we aggregated some data there and presented at the Biostimulant World Congress. As an outcome from that, we're currently refining that data set, and particularly now that we've engaged with other stakeholders in the value chain, such as food multiples and insurance stakeholders, we're also looking to repeat the exercise with abiotic stress data. So we are going to spend a lot of next year engaging with the value chain, just to understand their data quality needs in that respect, so that we can tailor a data collection tool that's fit for purpose, that can address their data needs in that regard, in terms of compliance and underwriting risk. And then what our hope is then is in 2026 that we can look to start collecting data in this regard to increase the credibility and support the adoption of biostimulants and increase market growth. So to sum up, I know I'm kind of caught in time here, EBIC's journey is marked by several achievements. I think we've helped build a credible foundation for the industry, from regulatory wins such as the FPR. We now have just published in the last week SEN standards that support regulation. We have a code of conduct as a function of membership. So I think each of these milestones point to EBIC's commitment to responsible growth of the industry. And I think we've set the stage for continued market expansion and set a very strong foundation in that regard. So thank you very much for your attention. And I really do encourage you to engage with us, join our initiatives, and let's help shape a sustainable future for agriculture. Thank you.
Thank you, David. That was very interesting. Good work to promote high quality biostimulants. I have two questions, or sort of two parts of one question, and it's on the UK participation in ABIC. So do you have any companies, or can they join UK companies to ABIC? And do you have any UK policymakers or regulators approaching you or being part of ABIC?
Yes, membership is open to UK participants, and we do have currently a number of UK companies in our membership. I understand there is a contractor that's working with the regulators in the UK currently. They have made approaches to us because I think what we're doing with respect to the FBR and SENS standards. So there is engagement is the answer to the question. Yes.
Okay, very good. Thank you. I think that's it. So with that, we're going to move on quickly to Professor Ali Siah from UNIA in Lille, and he's going to talk more about the mode of action. So Ali, the electronic floor is yours.
Thank you, Matt. I think that's just able to share my screen.
Excellent. You can see that.
Well, so quickly, I will try to provide you a few information on the mode of action of biostimulants. Of course, biostimulants is an increasing research topic, as you can see here in this assessment of the new articles that have been released last years on the topic of biostimulants on plants. Actually, the number of research projects and also publications is really increasing since 10 years approximately. And actually, this increased interest on biostimulants is not only in Europe, but everywhere in the world. And this, of course, increase in terms of activities, in terms of experimentations, is really related to the current context of agriculture, and especially also on the future challenges related to, of course, climatic change in priority, but also to the lack of some few resources of fertilization, etc., in the soils. In terms of the activities of biostimulants, they have been, of course, introduced by, of course, the previous presentations. And here, just remember that, of course, biostimulants are really used to allow to the plants the use of resources that are available in the soil, but also especially for the adaptation to the constraints that plants can find in the environment and especially in the soil. Here I mentioned just some of course, constraints, but there is others like salinity, like other environmental not suitable conditions. But here I mentioned that's the most relevant one and the most problematic one for us in Europe. The activities of biostimulants have been introduced. Here, just again, perhaps summarize the functions of the biostimulants. Actually, the the objective here is just to mention that, of course, the mode of action of the biostimulants depends on the functions they provide to the plants. And of course, there is a few functions related to the nutrition to, of course, optimize the nutrition for the plants, for the use of the available nutrients, but also to release the nutrients that are not available in the soil. And this kinds of activities is mainly linked to microorganisms, for example, that can release, for example, iron and phosphorus in the soil to help the plants for nutrition. And also we can have biostimulants that allow to the plant tolerance to different stresses, abiotic especially, and also the increase of the quality, of course, of the yield, etc., and the quality of the quantity also of the yield. In terms of categories of biostimulants, as you can see here, we can find different kinds of biostimulants in the market, but also in the literature. There is different, classifications, but I took this one, which is perhaps the most easy to follow, and also perhaps related to three main categories clearly distinguished. We can find, of course, the non-microbial biostimulants, which contain, for example, protein hydrolysate, seaweed extracts, humic and fulvic acids, amino acids, and also other microbial, for example, biostimulants, like, for example, mineral biostimulants, something like this. And also we can find here microbial ones based on microorganisms like mycorrhizal fungi, azotobacter, rhizobium, azospirillum also. So these biostimulants are based on living organisms, and their interaction with the plant is really important to consider to understand better their mode of action, actually. And also, finally, we can consider the waste-derived biostimulants coming from, for example, byproducts from industry, from agricultural byproducts and food waste, etc., which consider, in terms of circular economy, these kinds of products take place more today in the market of biostimulants. And of course, what is really important to have in our mind is that this product could be, of course, formulated. This is the case of the product available in the market, but also sometimes not formulated and tested on the plants without formulation. And in this case, we can talk about the mode of action of the basic substance. The active substances, which are the main component of the product. And when we check actually the mode of action of the biostimulants in the literature and publications, we have really complexity in terms of the mode of action because of different reasons. Because sometimes we can find products with a mixture of molecules. This is the case, for example, for seaweed extracts, where we can find, of course, polysaccharides, perhaps sometimes hormones, sometimes also minerals, etc. And also the complexity is that sometimes we have, of course, the activity that depends, for example, on the varieties of crops on which we test the molecules, the environmental conditions. And in this way, we can have really complexity in terms of the characterization of the mode of action of the compounds. But we have some information which are available for some products. And here I just mentioned that some products of the biostimulants, of course, can be recepted by the plant with specific receptors. And in this case, we can imagine early responses in the plant cells that lead to, of course, reactions in terms of physiology that can help the plant for, of course, tolerance to abiotic stress, and also sometimes to, of course, just increase the growth and the development of the plants. And in terms of the main activities and also mechanisms that the biostimulants apply on the plant. Here I just summarize, for example, the main and the most frequently found ones in the literature. Here you have the biostimulant applications on the plants that can be, of course, done by foliar treatments or by soil application. And in this way, we can, of course, observe different effects on the plant. On first, the molecular level here, that can be, of course, like gene expression, regulation of different genes, and also the regulation of transporters of nutrients for the nutrition of the plant, and also a regulation of some genes involved in the stress adaptation, actually abiotic stress. And at the cellular level, we can observe increased photosynthetic activity, chlorophyll content, pigment content, secondary metabolism regulation, and also an increased enzymatic activity that helps the plant to reduce the toxicity of the abiotic stress. And finally, we can, of course, observe a different phenotype traits on the plants, like improving the roots and also the shoots, yield, etc. And this generally phenotype observations are really the result of the molecular and the cellular changes that have been induced by the biostimulants. Perhaps to give an example of the different mode of action that we can observe on the plant after biostimulant applications. Here we take into example, for example, the humic acids that when are applied to the plants, we can, for example, observe an increase of the root size, of the content of the sugars, photosynthetic pigments, total phenols, flavonoids, and also an increase of the antioxidant activity. And also in terms of antioxidant activity, the enzymes related to this activity can, of course, reduce the ROS concentrations in the plants, which are harmful for the plant after stress, application. And also we can consider, for example, the decrease of stimulatal conductance leading to a decrease of transpiration to save water in the plant. We can imagine, of course, effects on, protection against DNA hypomethylation and damage caused by the stresses, and also the regulation of genes involved in, heat adaptation, like heat shock proteins encoding genes, and also finally, for example, the regulation of genes related to water transport, nutrient absorption, and also the transfer of, for example, nitrates from the soil to the roots. To provide a specific example for, the biostimulant, how would they work on the plants? I give this example of best A, which is phytosterol-based biostimulants, because actually the phytosterols are natural molecules that are present naturally in the cytoplasmic membranes of plants. And there is today in the market this product marketed by this company, Illicit Plant, that when applied on, for example, some crops like maize, we have an increase of the yield. And actually, this increase due to two main mechanisms, which the first one is the reducing of the evapotranspiration. This effect is linked to stomatal closure that can, of course, limit the dehydration of the plant. and also the increase of the root developments within the soil that help the plant to optimize water resources, and also sometimes the farmer to do economy of water use. And of course, these phytosterols interact mainly in terms of activity with the plant membranes, where that can stimulate then later the mechanisms related to these activities. Of course, when we check the effect of biostimulants of the plants, we can of course observe different effects, like the plant growth and development, and also tolerance to abiotic stress, here in blue, of course. And also what we can also find sometimes is that when we apply biostimulants on plants, we can also observe an increased adaptation to a biotic stress. This is what have been discussed before. And in this case, of course, we can imagine, of course, molecules or microorganisms that's doing all these, of course, activities together. And when we, of course, want to understand better how this function on the plant, it is sometimes complex, and we have really lack of information about what happens when we have multi-stress on the plant. Actually, when we have a biotic versus abiotic stress, for example. This, of course, issue have been discussed several times in literature. As we can see here, that's when we apply, of course, an abiotic stress on plant. Of course, we can regulate different physiological pathways, especially those here related to abscisic acid, for example, pathway that lead to, of course, modifications in the physiology of the plant, leading to, for example, the production of PR proteins, which are, of course, phenylpropanoid-based molecules. And in this way, we can also observe, of course, the resistance for the plant to biotic stress. And this means that when we apply biostimulants miming, of course, abiotic stress, we can lead to tolerance to biotic stress. This is really a common mechanism that we can find in the literature because of this crosstalk between different pathways of the plant, as illustrated here in the right, where we can find, of course, the application of different stresses or perhaps biostimulants that mimicking the stresses. And in this case, we can observe of course, receptors that can, of course, be specific to one or both stresses. And then we have, different mechanisms and also cascades of reactions that lead to the tolerance resistance to both stresses sometimes. Perhaps quickly to conclude my presentation, I mentioned that we can, of course, to study the mode of action, we can use different methodologies, and more we can, of course, concern the methodologies and focus the methodologies on the process in phenotyping, for example, with using result boxes, as we can see here. To, of course, determine with the precision the effects of the biostimulants on the root development. And also we can imagine the use of different physical, of course, methodologies, like, for example, the use of imaging based cameras for for example, thermal cameras, or for example, the multispectral and hyperspectral imaging, and also the use of liquor technologies to follow easily of course, the photosynthesis of the plants and the accumulations of the pigments in the plants. These, of course, techniques and technologies can help, of course, the companies and the researchers to better understand the mode of action of the biostimulants. And finally, of course, we can use different omics approaches, like here, the use of the transcriptomic to have a global overview on the effect of the biostimulants on the plants. Of course, proteomics to check the effects on the proteome after the biostimulant applications. And also we can imagine the metabolomics, which is really a new, interesting techniques to better understand the effect of the biostimulants. And all these molecular, of course, techniques, which are not targeted techniques, could perhaps better answer to a few questions that, of course, companies or researchers ask when they apply the biostimulants on the plants. The time is over. I think that perhaps just to quickly conclude, I wanted just to mention that, of course, in the market and literature, we have a wide range of categories of the biostimulants. And of course, we have actually a complex mode of action because of the presence of mixtures of compounds in the products we test. This means, of course, that we don't have a really generic mode of action for the biostimulants and no common mode of action for all biostimulants or categories of biostimulants. We can here conclude that, of course, the mode of action is at least product dependent and sometimes perhaps condition dependent. And of course, the environmental conditions can also change the mode of action of the biostimulants in case of field experimentations. Of course, we can imagine also a crosstalk between responses to biotic and abiotic stresses in the plants. And of course, the question is really relevant and perhaps introduced by Patrick about the regulation among biostimulants and biocontrol compounds. And this is due to this, of course, common pathways that can naturally occur in the cells of the plants. And this is really a key question that's perhaps in the future treat with, of course, scientific issues. And perhaps also mention that in the future, it will be really interesting if we perhaps make more efforts to characterize the mode of action of the biostimulants, perhaps by using new methodologies, more sensitive, more accurate. And of course, the biostimulant market still perhaps poor in terms of innovation, and perhaps in the future it will be really relevant to make efforts to identify new products with perhaps a new innovative mode of action, and perhaps in this way we can imagine, of course, more efficient products for the farmers. Thank you for your attention.
Thank you, Ali. That was really good. Some solid evidence for mode of action and for how complex it actually is. So when you spray for avoiding, let's say, drought stress, you might actually be increasing protection against pest diseases at the same time. And this was one of the questions which I think you addressed quite well, where the advisors have different products that can advise that do not claim directly, but it's known they have an effect on biocontrol or control of pest diseases. And yes, that can happen. In interest of time, I'm going to quickly go next to Els. She's going to talk, she's from Viaverda, and she's going to talk about more, shall we say, product-based presentation of different products and her experience across different diversity of biostimulants. So Els, take it away.
Thank you, Matt. Can you see my screen?
Yes, all good.
Okay. Well, good afternoon, everybody. Indeed, I'm working at Viaverda, an institution, and I'm responsible for all outdoor crops, so vegetables, ornamentals, and potatoes for the research. And I want to tell you a little bit more about the different types of biostimulants that exist. Ali already showed a way to categorize these biostimulants. I use the categorizing that we used in the Bio4Safe project, and which is also the base of our database, which we'll tell Matt more about later. So we have 6 major categories of biostimulants. The first one is the humic and fulvic acids, then the seaweed and plant extracts, then the protein hydrolysates, then the chitosan, the inorganic compounds, and the microbial-inoculants. Yeah, we can put them into their category, but there are always some biostimulants which are a combination of different categories. For instance, here I have two examples put there, which are combinations. So then it's difficult to say, oh, okay, we have this effect because of, for instance, seaweed extract or because of amino acids, if they are combinations. So they can't be put it all into one category. But I will start with the first category, which are the humic and fulvic acids. And in fact, they are one group, but there is a big difference between them. You have a humic acid molecule and the fulvic acid molecule. And if you look very well to them, then you will see that the humic acid is much far bigger than the fulvic acid molecule. And that means that there is also a very big difference in mode of action, or what can we expect of these products. The humic acids, in fact, they are insoluble in acidic environments and soluble in alkaline environments. And that means because of, and they have, of course, because they are so big, a high molecular weight. That means that insoluble in acidic environment, that maybe it is hard for them. If we test them on very sour environments, it can be hard for them to do their actions. While fulvic acids, for instance, they are soluble in acidic and alkaline environments because they have a low molecular weight. Maybe they can work more easy in acidic environments. Another component that is made in the same production cycle are the humins. These are insoluble and are in fact difficult to have a mode of action because of their insolubility. So I must admit, I did a lot of trials with Humifers, for instance, or something, a product, biostimulants that contain a lot of humic acids. And before I did more than 20 years research on sour-loving plants like azalea, rhododendron, and so on, in very sour environments. So maybe I choose the wrong product. I should maybe have used fulvic acids to test it because they were acidic environments, and their humic acids have more difficulties to be soluble than fulvic acids. So therefore, my message is, we really have to know the mode of action. We really have to know the products we're testing in to make the right choice. For instance, the Humifers, it contains more humic acids than fulvic acids, while fulvic, this product, contains more fulvic acids than humic acids. Before, I never looked into it into detail, but it is really important to do this. And if it's not indicated on the bottle or anywhere, the grower really has the opportunity to ask it to their supplier. They must give that information. So really ask what is in it and what can we really expect of it. Another action that we can expect from humic and fulvic acids is that they can bind with heavy metals. And they also can improve tolerance to abiotic stress because they can produce polyphenols, which can mean that they can cope with salt stress and drought tolerance. And also because of these humic acids are so big, maybe they are mostly or better used in soils, while fulvic acids can also be used in smaller substrates or container pots or something. We did already some trials with humic acids, as I said, in pots results were not always good, but I will show later on a result in full ground where we had better results. The second one are the seaweeds and the plant extracts. Well, seaweeds are the most common used and are the ones that you will find most, are the biggest group, in fact. And the one that is really known most is Ascophyllum nodosum. That's a very, very popular seaweed. It looks like this. And there are really a lot of products containing this Ascophyllum nodosum. I don't show them all, but there are really a lot. There were already questions about it. Yeah, they also contain a wide range of natural plant regulators like cytokines, auxins, gibberellins, and therefore they, of course, promote growth and root development. But it's really a mixture, and that mixture sometimes can change depending on the product. So also there, it is important to know what is really the content of the product, and that can make a difference in their action. Another one is Eclonia, also that's also very known, and it has a high content of natural antioxidants. Also, different products contain Eclonia, and we did ourselves in the Bio4Safe project tests with kelp, with laminaria. That was also one of the questions in the chat. Also there you have a lot of possibilities and a lot of products. And also there are other seaweeds that can help and are recognized as authorized, as biostimulants. To be honest, they prove a lot of literature and so on prove that seaweeds can have a good effect to to cope with drought stress and so on. But in our trials, it's always very, very hard to prove this. The effects here, as they are listed, you can see there are a lot of effects, positive effects that these seaweeds can have, but it's so hard to demonstrate these effects in trials as we perform when mostly conditions are already very good. Okay, we move to creating stress conditions, because without stress conditions, we certainly can't see anything. But even with stress conditions, it's difficult to have the right stress and not 100 other stresses in your trial, and to know to what point will I go with my stress. And if plants are not totally homogeneous, then the one plant will be over the point of no return, while the other plants, for instance, are still too less stressed. So it's really hard to prove these effects, certainly with seaweeds. Then we have the protein hydrolysates. The protein hydrolysates mostly that are mixtures of amino acids and peptides. Also there, these are a very important group. And you have the mixtures of the amino acids, and you have also the individual amino acids. There, it's very important for a grower to ask what is the source of the protein. You have those of the animal origin, and you have those of the plant origin. And also the method of the hydrolysis is also important. You have the more chemical hydrolysis, and you have the more enzymatic hydrolysis. Depending on the method and depending on the source, you can expect other effects. So also there, ask the supplier for this information. You have the right to know it, and it's important to know it. Yeah, that can explain why one product is working on your crop or not. But also here, there are a lot of positive effects promised, and there are also a lot of products that are available. I only mentioned some. We did also test different of these amino acids. on our trials, but again, there mostly against stress, and that was drought stress, and that was again very difficult to perform very good trials. We now have already a lot of experience of setting up trials, because that's also very important, setting up a very good trial, and that means that we know now which plants are good to test on drought stress, which conditions should we try to emphasize. But it remains far more difficult trials than, for instance, testing pesticides against aphids or something. With kite design, we don't have so much experience. It is only not only kite design. This group, it includes also polymers of biological origins, such as cellulose and starch and others. But in fact, kite design and chitin are not the same, but coming from the same products, the crushed shells mostly. It's just kite design is just another product in the production cycle. So it should work also against a lot of things. The most known is as antipathogenic agent also, so a bit the gray zone. But it should also enhance plant growth and development. And sometimes in very controlled atmospheres, you can have indeed a very good growth. For instance, here the root growth that is really affected by different chitin and chitosan doses. But also in our trials, again, it was so difficult to show this difference, and maybe it should be done in more controlled atmosphere then, but that's not realistic. If we go to the grower, there it is also not a controlled atmosphere. So also this group very hard to prove the efficacy, and kite design is not cheap also, so that's also a disadvantage. Then the next group, an important group also, are the inorganic compounds. These most known are silicium, aluminium, cobalt, selenium, and they promote plant growth and the quality of plant products, and also abiotic stress tolerance. Well, the most important one is the silicon, and most proven to work, and also most tested one. Yeah, you can imagine that it strengthens the cell wall because it improves the uptake of calcium and potassium. But it can also work on various way of stresses. But here also, again, there is something important. Silicium is present in the environment a lot, but it's mostly in this way, SiO2, silicium dioxide, in this form, and that's insoluble. It is, in fact, the orthosilicic acid that is soluble. So if you buy silicon-based product, then you have to look very carefully that it is orthosilicic acid, because it has to be soluble to be effective. And you can see that there are products on the market where it's only the silicium dioxide that is present. I would say prefer using products with orthosilicic acids to have effect. So there again, look at what's in it, and if it's not on the label, ask your supplier. Okay. In this group, there are also inorganic salts of beneficial and essential elements like chlorides, phosphates, phosphites, yes, of course, silicates and the carbonates. And we are there also again on the gray zone, because, for instance, I give one example. You have the phosphate, which is known as a fertilizer. Then you have the phosphonate, which is not a fertilizer, which is a pesticide like Aliet, that's phosphatyl aluminium. But you have the brother of alliate, potassium phosphide, which is not known as, not authorized as a pesticide, but falls in this group. But yeah, we know also that it can have a side effect, but that is a question that also Patrick already answered on it. So here you have also products on the gray zone. And then we come to the last group. The last group are the microbial inoculants, which is a very important group. The very important group containing the bacteria, containing mycorrhiza and trichoderma. First, the bacteria. There you have the nitrifying and the denitrifying bacteria, and this the nitrogen cycle, like you see here. Yeah, everybody knows we go how this cycle goes. So these bacteria play a role in the availability of nitrates or nitrogen to the plant, and are of course very important for it. And there you have, for instance, the nitrogen fixing bacteria. There are, in fact, many bacteria that can fix the nitrogen from the air. And you have three groups of them: the symbiotic bacteria, the diazotrophic bacteria, and the endophytic bacteria. Some of them I saw they are on the positive list that Patrick already told about, the CMC7, but not all of them. The first group, the symbiotic bacteria, Rhizobium, there you have also products that have this bacteria in it. They form root nodules, which can fix then nitrogen. The next group, there also many products contain, for instance, Azotobacter. They can also fix nitrogen when oxygen is, for instance, low. And then endophytic bacteria, like Utrecia or blue stem, I did also test this on it, and they can also collect nitrogen from the air and make it available for the plant. Our test, for instance, with this product was with plants that were really stressed. They were really in a low nitrogen level. They had problems with the roots, so there they could not take very good the nitrogen. And I could save the plants thanks to, for instance, spraying this product. But again, it was in these conditions that it worked very well, which will not mean that it will work in all conditions, and it will not mean that it will work for all plants. So it's so dependent, and that makes it so difficult. But sometimes it can be really helpful. Then another type of bacteria are the phosphorus solubilizing bacteria, that are bacterial strains that are that have the ability to solubilize insoluble inorganic phosphate that is common in our environment, and they can solubilize it. We did also tests already to try to have this effect, but also there it was difficult and we could not really show big effects. But we should repeat and test it in other conditions. And then you have indeed, also that was a question in the Q&A, phytohormone productions, bacteria that can do that can produce plant growing, promoting rhizobacteria, like the hormones like auxins, cytokinins, gibberellins. Yeah, and when we hear, for instance, auxins, then we think of better rooting. And indeed, these products are known as they can improve rooting. We did tests on cuttings where we saw really a positive effect. And I also did already with these kind of products trials at grower sites. where a grower had really problems, for instance, with his plants, and we could also improve the effect of, we could improve the rooting. So when there is stress, then it can be helpful. When everything is really okay, then it is hard and almost impossible to see the differences. Yeah, and that's a really very, very complicated way and mechanism. Ali already told us some about it, but I will leave it there because it is so complex, which can be the reason why it's also so complex to show the differences or to show the effects. It depends the plant, it depends on the environment, it depends on a lot of things. And then the last group, the second group are the mycorrhiza. Mycorrhiza are also known that they can help plants. They live in symbiosis with the plant. So both the mycorrhiza and the plant has an advantage of living together. And they also have a lot of positive effects. They are already known to have, and a lot of research is already done on these mycorrhizal fungi. And of course, a lot of products are available in this category. And then the last one are the trichoderma. I also saw that people are using it. Trichoderma, which can interact it with the roots, a bit different as the mycorrhiza, but they can help for instance, having bigger roots and longer roots. They colonize the rhizosphere, and they in fact compete with other organisms, and so they can improve plant growth. There are a lot of effects that are written to them, but the most important ones are the plant growth promotion and the root development promotion. Also here you have trichoderma that are authorized as plant pesticides, like for instance Trianum or others. That's a Trichoderma harzianum. But that's a very specific then trichoderma known as being a pesticide against a fungi. While here you have a group of trichoderma, put it together in one product. So not specified this or that trichoderma and not against a specific pest. So it's different, but I understand the gray zone. Then some small trial, it were big trials, but very shortly about some trials that we did together with some students. For instance, here it was a trial on Hydra, Spathiphyllum, Viola, and Primula, where we tested different products, and we looked to rooting was very important, but also plant quality, the plant height, and the diameter. The advantage of these products is that you don't have to, most of them don't have to applicate many times, mostly, for instance, after potting or during potting, or for instance, just after potting and 2 weeks later. But there were also products that you really have to apply every two weeks. And for some growers that can be doable because they have a sort of system where they have to use other, they have to spray with other products, or they can put it in their water and give it in the system. But if you really have to do the spraying every two weeks as a grower, the suppliers must be aware that that's really a bottleneck. That's not easy for a grower to do. And the cost, of course, of the product is then also very high. But we had some positive effects of two of the products that we tested. And we had some more negative effects of these two products, but the same as Patrick already told in his talk, in other trials with the same products, we had positive effects. So you can have really from these plants in these conditions this effect, on that plant other effects. So therefore it's important that we have the time and the money to do a lot of trials, because I want to make a statement also. The European Commission is really dispensing a lot of pesticides. It's diminishing harder and harder. They think that biostimulants will save us, but if this that's the case, then the European Commission should give more money to do these trials, so that we can help the growers to check whether they can make plants more stronger, so that they can survive pests and insects and so on. Another trial on salvia, pelargonium, rose and cordyline with other products that we tested. Here we had to spray really a lot, which was hard labor, I must say. That's really something, like I said already, to take care of. But there also we had some positive effects, some, for instance, on this product, then more another product, 2 other products gave at a certain moment bad effect, but then afterwards, then again, a good effect. So it really depends on the plant, which effect you can have. And then we have the trial on acer, which was performed in the project Ornaqua, also at Viaverda. And there, like I said in the beginning of the talk, Univerisity gave a very good effect, together with a biostimulant that we tested, which is not commercially available already, and a wetting agent. So we can have good results in full ground, as I said, with Univerisity, which I did not have in pots. And then thanks to Réfléchis, an Interreg France, Walloon, Flanders project, we did some trials at Viaverda, Kruisem on onions. There we tested these products, in fact, and we saw positive effects of Hamerol and Acticil, and certainly in yield we saw also a rather big positive effect of Acticil. We have shown these data to growers, and other growers were talking about a good effect of Afazol. I saw it also in the Mentimeter doodle we did, appearing that people are using these products. So just to conclude, there are products that give positive effects. We can't say that it's all the same for the different crops and for the different situations. But there is a kind of, I can come to kind of conclusion. It is in fact a translation of a publication of the WUR, and you can just find it on the internet. And there they tried to put the different effects and which category we can expect to do this effect. For instance, when we have, and they divide it in the microbial group and the non-microbial group. And for instance, if we take, for instance, the competition for food and space with pathogens, then it's good to test bacteria. If you want some changes in soil culture structure, there also the bacteria will help and the mycorrhiza not. The same for increased physical defense. And also for the non-microbial group, you have these effects like let's just say the activation of specific plant genes. Well, you can then look into the group of the plant and seaweed extracts, the humic and fulvic acids, and the nutrient polymers, and so on. For all the different mechanisms, you can go into the right subcategory. So always look at the label. It's very important to do it. And then look at our database, which Matt will tell you more about.
Okay, thank you very much, Els. That was very good from very experienced researcher to see that there are some positive results out there. I have a couple of questions here, and I can address the first one probably on phosphates, high phosphates and high nitrogen inhibit mycorrhizal associations. Yes, they definitely do. So in over-fertilized fields or very highly fertilized fields, it's very difficult to get mycorrhiza established because the plants will reject or will not form symbiosis with mycorrhiza. So you're basically going to be throwing inoculum out. There's very little association. So on those fields, it's probably recommended to reduce the nitrate and phosphate levels by maybe not fertilize it for a couple of years before go to AMF and then keep it. Then mycorrhiza will keep there better. And then here, one for probably all the speakers is that one of our attendees is saying that it would be nice if the biostimulant tests and results always include references to the feed, soil type and fertilizer use, because they do not. So having standard rules would be very good to make sure that the biostimulants could be used better if they're tested and then the test results or the conditions of the test results are known so that they can be replicated. So maybe David has a hand up that can respond to that.
Yeah, thank you for that question. In the last week, standards have been published which support the fertilizing product regulation. So these standards, one of the standards has to do with general principles. So that goes into specifics with regards to experimental design, data quality, statistical assessment, your technical replicates, plot sizes, and things like that. And then for each of the functional claims, such as nutrient use efficiency, there's about 6 indices that are included in terms of how you measure use efficiency. Equally then for abiotic stress, data quality, and increasing the bioavailability of nutrients in the soil. There are standards that kind of govern all these areas. So these will help standardize the data coming from trials that will feed into the claims. So there is dimensions being put on it to address this. And I suppose the other point I would say is we also encourage peer review standards. So with the Biostimulant World Congress, there is an editorial gateway there as well in terms of feeding in state-of-the-art or new state-of-the-art data with respect to the efficacy of biostimulants and their mode of action. And the reason we want to do this and be a part of this is to ensure that biostimulants are grounded in science, that there is a characterization that goes from the composition and looks to build relationship with function within the plants. That is important for steering the industry forward and giving confidence in products. So there is, because I came from industry, I spent 14 years in the seaweed industry. There is a new wave of biostimulants coming that are kind of the next generation as well, where some of those relationships have been more pinned down and will give stronger efficacy. So that's important to point out that the FBR really sets a baseline. It's a limit. It's the start, not the end. So we do encourage our members, and a lot of them are very active in terms of R&D, to build that data set and that confidence in the products by underpinning it with solid science.
Okay, brilliant. Thank you for this. Thank you for your contribution, David. We have a few more questions we're gonna answer at the end. And in the interest of time, I will now share my presentation on the database that we've been building sort of since 2018-19. I hope you can see my presentation.
Yes.
Very good. So I'll just jump into it so we don't get too over. Yeah, there we go. So the very first thing first, it's an online database. You can find it on the address on the top, or if you just search bio for safe with 4 as a number, you'll find it. And we actually had a very specific aim for this, and that is to combine all available and reliable published research on all relevant commercial biostimulants, and that's relevant for Europe. And why we did this is to aid the selection of products and product groups like seaweed extracts or microbes or AMF or other groups for growers and end users primarily. And we wanted to show all the data that is out there, which products have worked, which products have not, on which crops have worked and which not, and which conditions. So that's going to answer a few questions in the chat. And we want to disseminate this biostimulated research between academic and industry spheres to connect them a bit better. We started this, as I said, in not 1984, 2018 is when we started this, but we included research in the first iteration that was under EU Bio4Safe project, had research from 1984 to 2020 that was included in that first database. And now we have updated it from 2020 to 2024. So we're up to date with most of the published literature should be in a database. So it's free available online resource. And the idea was that it's quite tricky for everybody to understand what the biostimulants research is about, or what it was done and how it was done and what's the outcome. So we try to distill outcomes of the research papers and trial data into clear and simple summaries. We have each summary is for one biostimulant product and for one crop. So if one biostimulant has been used on multiple crops, we have multiple entries. If same crop has been used with multiple biostimulants, that's different entries as well. And we wanted to make it as user-friendly as possible, to be searchable and filterable. Now, we're not a very high tech, we're sort of biology scientists, so we had a little help, and database may not be super fancy, but it does work. And what we really wanted to include is to give the power to growers to sort of investigate themselves a bit what they're doing. So we want experimental conditions to be included, application details to be included, whether it's spray, at what dose, at what time, and then give them the reference, and if they want more, they can go and look for it. And for all the researchers out there, we do have a large sort of raw data table with all the data that's online. It's in one massive Excel sheet that we can share on demand. We've done more than 290 peer-reviewed scientific papers, and we have included, after our own screening, trial reports from companies or other research institutes that had good experimental design, were commercially used, commercially available biostimulant products. So not something that they've distilled or extracted for the particular study, but something that was off the shelf with a product that was sold somewhere in some shop. And we reported all effects, whether it was positive, negative. If they had no effects, we always reported everything. And we stick to all the definitions of biostimulants, so you won't find anything that has biocontrol claims on it in our database. But we had to focus on something, so we focused on crops grown in Europe. So there is no, what I call here, lab bunnies, so no Arabidopsis or other plants that are not grown as crops. And we did not look at all the crops that are grown across the world. So there's a lot of very temperate or tropical crops that were not included because it was mainly focused on Europe. So at the moment we have 907 entries, and we're still updating it. We have over 130 different crops in and over 250 biostimulant products in. And as you can see on the picture here, pointer options. Oh, that was not good. I'm going to try pointer options. So you can subtitles. Stop subtitles. Very good. So you can search the database based on the effect that you want your biostimulant to have, based on the type of the biostimulant that Els was talking about and everybody else, or based on a crop that you have. And what we have in our, I just put some fancy word clouds here. We have loads of different crops, a lot of tomato, a lot of lettuce, a lot of brassicas, and for example, there's strawberry there. Also, all sorts of different crops you can find. A lot of cowpack, filgreen, and other seaweed extracts are prominent in the database because that's one of the main biostimulant that are sold biostimulant groups. And if you look at the effects that we report, there's chlorophyll has been measured, weight of either crop or plant, increased and decreased. You see here a lot. There's a fresh weight, dry weight, all sorts of things that is in it, but that is just like an illustration. And I will give you a few examples at the end. But what I wanted to say that there is more to the database than all the biostimulant data. And we have, through Bio4Sec project, we have used loads of sensors to measure biostimulant effects. And we put all the different leaflets on the sensors in 3 different languages under the sensor tab that you can find at the same page. And we have put all the trial protocols that we use to investigate biostimulant properties, biostimulant effects. We put here as well in 3 languages. So you have for growers and for researchers, leaflets and protocols if you want to reduce your water fertilizer use, which what we kind of focused in Bio4Safe, or if you want to know about what can you do to better monitor either of those, you can find these additional resources next to the biostimulant database. But just to give you an example, I'm not sure you can read this, and if you can't, we can just quickly go online and I will show you how it works online. For example, I looked for apple and increased quality, and I took the first 2 that came up. So there was ActiWave, and for ActiWave, we know that ActiWave applied to apple trees cultivar Fuji, grown without addition of NPK, had 50% higher shoot growth, 12% more chlorophyll, and decreased oscillations with on and off years, and so on. And you see that this was done in Italy, outdoors, was fertigation in irrigation. It was 30 liters per hectare. So you know how to use this product. If you see it, this is what the product has done. And if you want to read more, you see at the bottom the full reference. As another one here, for example, that was done at 2 liters per hectare as a foliar spray. And you can see that Gala apples treated had less nitrogen, more phosphorus, less potassium, and more calcium than untreated. And there was bigger in diameter and contained more dry matter. So we list everything that we could find in with percentage if we can give it to you, and whether it's significant or not, and how and when it was done. So that was the trial done in Hungary, and it's been published here. So you can have a look at this yourself, and I'm going to just try to quickly guide you through before I come back. So if I look at the database here, you can quickly search. For example, we're going to search for lettuce. No, something that I haven't searched for yet, so it's going to be a surprise for me as well. Let's say grapes. And I want to see, I'm going to leave effects. There's quite a lot of effects, so I'm just going to use for what we get for grapes. So we have loads of effects. So some have some of our entries have decreased effects, some increased, some have no effects. For example, I want to see in there's quite a lot of here. So let's see if I look at the cowpack in grapes, forage, strawberry, tomato, brown orchard. Oh, there's quite a lot here. I'm gonna have to limit my search, but let's assume that I'm looking for this one. Cowpack in spinach. If we have, so it increased product quality and decreased product quality depending on what you look. So we report whatever is there. The rate was 4% dilution foliar spray in South Africa, and they had more carotenoids, more chlorophyll, and lower leaf weight overall. So you can sort of know what to expect. And the aim here is not to give you like a choice, like a data that's going to say use this biostimulant rather than that one. The idea of the database is that you have information and you can then interrogate your suppliers, or as a researcher you can look for what are the other effects that have been shown for something that you investigate, and you can have a better idea. So with that, I'll invite you to have a look and search for yourself, and put any of the other questions in the Q&A box. And I would really like to thank all our past and current funders and collaborators and all the speakers today. I think they did a great job. It was very interesting. And I'm gonna pull QA up, and I invite everybody to join in. So Q&A, what have we not talked about? So just to say straight away, we will share the video links through our social media of all the institutes, and everybody who registered through Viaverda website should get a link. So that is that. I have a question here about wetting agents, and are they also considered a biostimulant?
We answered it, Matt. It is not a biostimulant, wetting agents, but Pauline already answered that question. I think it's only the two highest questions that we didn't answer yet. And that's the one, if we can, my screen is, if we would be able to add products in the database with, if there is a CE label, yes or no. Yeah, that's a difficult question. Maybe we should write a new project to be able to input all these data. It would be interesting. And also, for instance, where a question that I get often from growers is, can you put into the database also where a product is sold? Is it sold in Belgium or France, or which are available in which country? Unfortunately, this changes very quickly, and this is difficult to manage. In fact, it is better to look at the compounds of the biostimulants more than the names.
Yeah, I would say the same. I would say that we had a problem in Bio4Safe already, that there is no register of biostimulants in pretty much any of the countries. Maybe Belgium has some sort of register. So it's very difficult to know what is on the market. You cannot, for example, in the UK, you do not know, we do not know what is actually available on the market, and it changes year to year, and things come back that were not available a couple of years ago, and new things are coming constantly. So it'd be very difficult to include that in a database done sort of by researchers. There's something that should be kind of dictated from the top down. When you register a biostimulant, you'll have to sort of enter a list of things that are on the market in a particular country. But at the moment, we don't have that. So we can't really put that in at the moment.
I think then there are two more questions that we didn't answer yet. Will the database be updated, or will NIAP continue to update the database? In fact, we, the three partners together, are responsible for updating the database. Yes. Till the project is running, yes, we will certainly do it, and we are happy to work together. So we will look at new opportunities to be able to indeed update this database. That's the first question. And the second question is one I can't answer. I don't know if some of the colleagues know the answer. Do we have to consider risk to human health applying these microbial biostimulants? I don't know if somebody knows the answer on this.
I would say that very likely. The human risks to humans are come very, very early on in all sorts of products. Even in if you make compost, like for a bag for people to put plants in, you have to have a PAS 100 test. So I'm not sure if David has any contribution to this, but I certainly think that human safety is in all products on the market, probably.
Yeah. Just with the FPR, microbials are not yet on the market. There is a positive list there. Currently, there's a contractor that's being engaged to develop methodology, and part of their remit is to look at safety, not just for human safety, but also environmental safety and agronomic efficiency. So they really only started their work at the European level in January, and that won't conclude until December 2025. But that's for placing microbials under the new FPR regulation. But I would suspect at national level, as you rightly said, there has to be some safety standards that need to be met for placing such products in the market.
Mm-hmm. Yeah, so we can quickly discuss one or two more questions. One is, which criteria did we use to select the products? We did not select products ever. So we used all the research that was out there, including trial research that was volunteered by companies or research organizations, as long as they had high quality. And then we used all that to include into our database. So there was no selection. And just, yeah, I was just reminded that before you all leave, Pauline has a quick last question for you about the webinar and about the things presented. So I will let her quickly share that before.
Yes.
And you can still put questions in. We can still discuss for a bit longer.
Okay, thank you, Matt. So I still have 6 questions for you to close this webinar about your experiences with biostimulants if you already use them. So you can again scan the QR code with your smartphone if possible. And then we can start the small poll. The first question that you have for you is, do you have negative experiences with a specific types of biostimulants? So as you can see in the answers, we are using the groups that Els already explained in her presentation. So we have humic, fulvic acids, seaweed and plant extracts, protein hydrolysates, chitosan and biopolymers, inorganic compounds, bacterial and fungal inoculants, other, or the option, I'm not a grower. Because we know that there are a lot of not growers here in the audience. But if you are a grower and you already used a biostimulant and you had negative experience, you can answer it. Okay, some answers are coming in. Thank you for that. It's nice to see that. I will wait a bit because there are still 140 people here in this webinar. So I will wait a bit until we have more or less 100 answers. Most of the negative experience that you have come from using bacterial and fungal inoculants. Followed by humic and fulvic acids. Maybe because the other types aren't used that much in practice. That's possible. Okay. Then we can go to a next question. The other way around, do you have positive experience with a specific type or specific types of biostimulants? Because that's also interesting for us to know. Now we see in our own trials that we have most of the time no effect or even a negative effect. So it's also interesting to know if you have already seen positive effects when you're using kind of biostimulant on your company. So again, bacterial and fungal inoculants also had a lot of positive experiences. So that's surprising to see. The answers are coming in. Okay, then I will go to the next question. Did you saw no effects after using a specific type of biostimulants? Because that's something that we experience from our own trials. That's most of the times we didn't saw anything. So the bacteria and the fungal inoculants are not here, so they only gave positive or negative effects, but not no effects. Okay, there's an answer for the bacteria here. But it's more humic and fulvic acids and chitosans, biopolymers, seaweed and plant extracts that gave no effect. So in that case, it's not worth to buy them and to use them if they give no effect. But it will very depend on your crop, on the conditions where you will use them, of course. Okay, I think we can go to the next question. Are you thinking of testing new biostimulants at your company in the near future after attending this webinar or after reading some information online in magazines, things like that? So a lot of people are thinking of testing them and of using them on their company. So that means that there is still future for the biostimulants, I think. But also some people that are doubting. And I think we can then move on to our last question. No, it's not the last one, sorry. If you would test or use a new biostimulant, which type would you use after seeing presentations of today of Ali, of Els, where they described the effects of the several types? Are you interested in humic fulvic acids, seaweed plant extracts, or protein hydrolysates, chitos and biopolymers, inorganic compounds, bacterial and fungal inoculants, or other products? So here again, the bacteria are popular. So this is the second last question. The answers are still coming in, so thank you for that, for still being here with us. So after the presentations of today, people are most convinced of the bacteria and the fungal inoculants. And then normally the last question, how satisfied were you with this webinar? Did you find it are you very satisfied, neutral, unsatisfied, or very unsatisfied? You just can be honest, of course. Okay. Most people were at least satisfied and even very satisfied. So that it's nice for us to see that this webinar was interesting for you. Okay, then I will also stop this question and stop the poll. And then maybe we can still look at some questions in the Q&A if there is some time, if people will still attend. If you want to leave or you have to leave, you can do, but if you still have questions, you can ask them, of course.
Yeah, at the moment, we don't have any more questions, but thank you everybody who answered this last polls. It helps us to bit of a gauge, and maybe next time we should ask what would you improve, but that's something that is quite difficult to get from the audience at this kind of webinar events. But I'm glad most of the people enjoyed it. So I don't see any further questions. In which case, I think I'm going to conclude and thank all the speakers. Patrick had to leave already. Thank you to Barton, Ali, Pauline, and Els. I think it was a great webinar all around. I've learned a lot. And our
Thank you
participants.
too, Matt,
Yeah,
on
indeed.
behalf of the others. And I indeed think we can close this session. And hope to see or hear you in another event, I guess.
Yes. Well,
Yes,
thank you,
indeed.
everybody.
Thank you
I
very
will-
much. Yeah.
I'll be the last to leave, so because I think when I leave, it will shut down. I'm gonna
Yes.
Okay.
wait a bit more.